Hi, good evening, everybody. This is in continuation with today's previous lecture. Now, historically, the food industry in the United States has argued against labeling genetically modified foods, and because will be perceived as a warning and will unnecessarily stigmatize their products. On the other hand, as a marketing strategy, some food companies and retail outlets are specifically labeling products that do not have any genetically modified ingredients. Since a label cannot be misleading or untruthful, the FDA has objected to descriptions, such as no genetically modified organisms or genetically modified free, because in the former case, the product does not contain a viable organism, and in the latter case, there is no guarantee that some contamination has not occurred. Terms such as genetically engineered and made through biotechnology are unacceptable to the FDA when there is no need to specify nutritional or health requirements. Consumer advocate groups in the United States have argued that the consumer has a right to know if a product comes from a genetically modified organism, regardless of an equivalence of properties. Although legislation supporting this principle has been drafted by members of the U.S. Senate and House of Representatives, none of these proposals have had such or much success. Concerns about the impact of genetically modified organisms on the environment. There are currently more than 6.7 billion people on Earth, as this is as for the fourth edition that was taken. Now it should be much more. And the population is projected to grow to 9 billion in about 30 years, which I think is almost close or crossed. This is an enormous number of people to feed with an agriculture system that is already burdened by crop reductions due to disease, pests, adverse weather, droughts, flooding, and hurricanes, competition with non-food crops and soil quality deterioration due to overuse of agricultural chemicals. Production of transgenic crops can offer solutions to some of these environmental problems by increasing the resistance of important crop plants to infectious disease, and insect predation, climate change, and reducing applications of pesticides. However, some critics believe that our increasing dependence on genetically engineered crops is adversely affecting the environment by decreasing biodiversity and harming unintended organisms. Impact on biodiversity. In general, agriculture has decreased biodiversity. Crop plants are selected for high yields of the edible parts, hardiness, and other traits at the cost of loss of richness, not only of cultivated species, but also wild plants as cultivation is expanded over a great area. Newer, higher yielding varieties have tended to replace traditional crops. The diversity of insects, birds, and other animals has also decreased as more land is dedicated to agriculture and host and food plants are lost. Application of agriculture chemicals to eradicate weeds and pests is a contributing factor. There is some concern that the trend toward extensive cultivation of crops that have been genetically engineered to resist environmental factors that normally limit plant growth, that is, herbicide tolerance and insect resistance, could further decrease biodiversity. This concern stems mainly from the possibility that the transgenic plants could become weedy or invasive of natural habitats, or that the transgenes can be transferred from genetically engineered crops to wild relatives or to non-genetically engineered crops and could confer them on a selective advantage. Enhanced growth of the unintentionally engineered plants could cause them to be invasive or to outcompete plants without transgene. Several species of weeds have been found to be resistant to the herbicide glyphosate. For the most part, this is due to overuse of the herbicide, which selects for natural mutations that increase resistance in weeds rather than to the transfer of transgenes through crossbreeding hybridization, although the latter has been shown to occur at a low frequency. Pollination of wild relatives by genetically engineered crops can occur when they are within range of pollen distribution by wind, insects, etc., when pollen production by the genetically engineered crop and flower production by the wild relative occur at the same time. A transfer of genes among plants that mainly self-pollinate, pollination of a flower by its own pollen, such as rice and soybeans, occurs at a low frequency. For example, transfer of a herbicide resistance gene from genetically engineered rice to wild red rice was found to occur at a frequency of less than 1%. The frequency of transfer is higher for plants that cross-pollinate. The pollen from one plant fertilizes the ovule of another plant. The glyphosate resistance gene from transgenic canola, Brassica napus, an important oilseed crop, was transferred to a wild relative, Brassica rapa, or field mustard, at a frequency ranging from 7 to 14%, depending on the bee rapa population. This was determined by collecting seeds from herbicide-sensitive bee rapa plants that has been interplanted with the herbicide-tolerant transgenic bee napus, and then assessing herbicide tolerance and production of the specific protein encoded by the transgene in the plants that developed from the bee rapa seeds. While cross-pollination occurred at a relatively high frequency between bee napus and bee rapa, hybridization between bee napus and 3 other wild relatives was extremely rare. The spread of herbicide resistance gene reduces the ability of farmers to control weeds, and the weedy plants have the potential to become invasive as they can proliferate in areas where the herbicides are used. Herbicide-resistant plants are unlikely to have an advantage in non-agricultural areas where herbicides are not applied. Moreover, hybridization does not always confer an advantage on the hybrid progeny. Hybrids resulting from cross-pollination of transgenic rice resistant to glyphosate, a herbicide that disrupts glutamine biosynthesis, were less fit than wild-type parents because flowering occurred too late to produce seed, which prevented proliferation of the hybrids. Hybrids that express a beta toxin transgene may have a significant advantage when insect pressure is high. The potential for gene flow to non-transgenic cultivars and wild relatives and its consequences are included in risk assessments required by regulatory agencies before a genetically engineered crop is commercialized, and strategies to manage the risks must be in place prior to cultivation. The assessment considers the mechanism by which the plant reproduces and pollen is disseminated, the nature of the transgene at any selective advantage that may emerge, and the geographic context in which the crop will be grown, which includes the identification of sexually compatible wild relatives. Management strategies can include avoiding cultivation of a transgenic crop in areas where indigenous wild varieties are found, and controlling embryo and or seed viability through genetic engineering. The impact of the Bt toxin on non-target insects. In May 1999, a report entitled Transgenic Pollen Harms Monarch Larvae by Lossi et al. Was published in scientific journal Nature. In this study, pollen from Bt corn was sprinkled on milkweed leaves. The source was a food for the larvae of the monarch butterfly. An extremely high mortality was observed after monarch butterfly larvae fed on the treated leaves. The author concluded that extensive Bt crop acreage could have profound implications for the conservation of monarch butterflies. The study, which was conducted in the laboratory rather than under natural field conditions, incited much criticism. Scientists point out that appropriate controls were not used, the amount of pollen on the leaves was not calculated, and there was no indication whether the time of pollen shedding coincided with the feeding period of monarch butterfly larvae. These technical concerns were largely ignored because the monarch butterfly has iconic and aesthetic status in North America. Consequently, the study was widely reported, and the threat to the monarch butterfly formed the cornerstone of the campaign against biotechnology in general. However, after 2 years of detailed study, the initial skepticism of most entomologists was confirmed when it was proved that the risk of Bt toxin toxicity to the larvae of the monarch butterfly was negligible. Anti-biotechnology factions have been reluctant to accept these findings. Much of the literature continues to stress that Bt crops are a threat to the survival of the monarch butterfly. More recently, reports in the popular media have emerged suggesting that pollen containing the Bt toxin was responsible for severe reductions in the honeybee population in North America and Europe. Declines in the honeybee population, a phenomenon known as colony collapse disorder, were occurring at an alarming rate, and the consequences would be very serious if pollinators were not available to produce fruits. However, an analysis of 25 independent studies concluded that pollen containing the KORAH proteins that are present in transgenic crops currently under cultivation and that are effective against coleopteran and lepidopteran predators are not toxic to honeybees, a hymenopteran insect. After a great deal of investigation, the general consensus among researchers is that because the BD toxins are highly specific for target insects and are confined to plant tissues, the impact on non-target insects is minimal. Several studies have shown that insect diversity and abundance have actually increased the cultivation of BD crops compared to application of chemical insecticide sprays that have a broader target range. Environmental benefits of genetically modified organisms. Cultivation of plants that produce heterologous proteins that provide protection from pests has led to a global reduction in the application of agricultural chemicals, many of which are toxic effects on the environment. Spraying of insecticides was reduced by 94.5 million per kg, or 19.4% from 1996 to 2005 due to the cultivation of resistant cotton alone. Insect-resistant corn accounted for a further 4.6% reduction. For example, the western corn rootworm is a devastating and pervasive insect pest of corn and causes huge economic losses in the United States and elsewhere. Because the predator is difficult to control, farmers routinely apply insecticides to their cornfields before infestation by the worm is apparent. The cultivation of genetically engineered varieties of corn that produce the KORAH-BIOTECH toxin, which is effective against the western corn rootworm, have obviated the spraying of insecticides against that insect. Excuse me. Although some would argue that the widespread cultivation of herbicide-tolerant crops has increased our dependence on herbicides, it has actually resulted in a 25 to 30% reduction in herbicide applications to transgenic crops compared to conventional crops, or the use of glyphosate, the herbicide to which most crops are engineered to be tolerant, has increased sharply. Over the last decade, the use of other herbicides has decreased. The herbicides that are applied are generally at lower strength and are less toxic. In addition, the herbicides can be applied to fields containing tolerant crops later in the season after the crops have grown. This is dramatically reduced and on many farms eliminated soil tilling, the practice of turning the soil to remove weeds, and has thereby increased soil quality by reducing erosion, organic carbon loss, and has decreased fuel consumption by farm machinery. The latter benefit is not trivial in its impact on the environment, as the estimated reduction in emissions of carbon dioxide, a greenhouse gas, as a consequence of growing herbicide-tolerant soybeans and canola, is equivalent of removing 400,000 cars from the road for 1 year. Because different agricultural chemicals have different environmental toxicities, a better measure of their impact on the environment takes into consideration not only the amount of active ingredient applied, but also the toxicity of the chemicals to farm workers, the consumer, and on organisms such as birds and insects. Its persistence in the soil and its potential to leach into groundwater. These parameters are used to determine the relative environmental impact of each pesticide. Using these values, the global reduction in environmental impact resulting from changes in herbicide pesticide applications to genetically modified crops was determined to be 15.3% over a 10-year period from 1996 to 2005. Economic issues. Who benefits from molecular biotechnology? Developers of biotechnology products that have applications in healthcare, agriculture, industry have benefited financially from recombinant DNA technology. Global sales of all biotechnology goods and services by biotechnology companies exceed $70 billion annually, with firms in the United States accounting for more than half of the sales. Most of these companies develop pharmaceuticals, for example, in the United States and China. Sales from health products account for 87 and 77% of total sales, respectively. Recombinant pharmaceutical products that are highly successful include the therapeutic proteins erythropoietin and human insulin. which respectively have sales of more than 8.8 billion and 5.3 billion annually. Many smaller developers complain that the high cost of developing and commercializing a genetically engineered product prevents all but large multinational companies from profiting. For example, in the agriculture, biotechnology sector, where cost of developing a genetically engineered product can exceed $20 million due in part to stringent regulatory requirements for companies own or co-own about 80% of the genetically engineered products that have received commercial approval. DuPont, US Monsanto, US Bayer Crop Science, Germany, and Syngenta, Switzerland. This means that the recombinant drugs that could target rare diseases that are found predominantly in low income populations and transgenic crops other than herbicide tolerant and insecticide resistant crops, and those that are not widely planted, do not provide the revenues that are necessary to justify the high cost of product development. Improved varieties of crops that are important for resource-poor nations such as rice, cassava, and millet have not been developed extensively. In addition, product development has been limited by biotechnology markets such as the European Union, which in turn influence development in farm countries that rely on export to these markets. Farmers in both developed and developing nations have perhaps been the greatest beneficiaries of recombinant DNA crop technology, not only due to increased yields from genetically engineered crops, but also from cost savings due to fewer applications of agriculture chemicals and to reduce labor and machinery costs. And cereals, oil seeds, and nuts contaminated with fungi are not suitable for sale due to increased risk of contamination with mycotoxins such as aflatoxin, carcinogenic toxin produced by aspergillus. The economic benefits due to reduced mycotoxin contamination are significant at $30 million. How do views about genetically engineered food affect trade? The exports of agriculture products from the United States to the European Union were valued at over $10 billion in 2008. However, differences in consumer acceptance and regulatory views have often made trade between the two regions difficult. In 1998, the European Union placed a moratorium on the importation of genetically modified corn, cotton, and soybean products. This prompted a complaint in 2003 by the US, Canada, and Argentina to the World Trade Organization, which ruled in 2008 that the additional risk assessments and approvals required by the European Union for some of the genetically engineered products were not based on scientific evidence and unfairly delayed trade. In contrast, the US In contrast to the US, only a few genetically engineered crops have been approved for cultivation and importation to the European Union to avoid labeling foods in more than 9.9% is derived from an approved genetically engineered ingredients. Exporters to the EU have been forced to separate genetically modified crops from conventional crops, which is costly. The resistance of the European Union to transgenic products has forced many developed countries to forego the benefits of planting genetically engineered crops for fear that trade with the European Union will be jeopardized. Summary. When assessing the acceptability of products of molecular biotechnology, people generally want to know what the benefits are compared to similar products derived from conventional technology, who benefits from the biotechnology products, and the risks associated with the products. In particular, consumers are concerned about the safety of foods from genetically engineered microbes, plants, and animals, and the impact of farming, genetically engineered crops and livestock on the environment, the attitudes of consumers, and rigorous regulatory requirements for commercialization have greatly influenced the development and availability of biotechnology products. Many countries, including the US, have adopted principles of substantial equivalence when evaluating the safety of genetically engineered foods. This means that a genetically modified plant or animal food product must be similar in composition to the corresponding conventional food. Levels of nutrients, anti-nutrients, and natural toxins must not be different, and animal feeding trials must not show differences in the development, health, or performance of the animals that will indicate reduced nutrition or increased toxicity or allergenicity of the genetically modified food. If the genetically engineered product is not substantially different from the conventional product, then labeling it as genetically engineered is not required. Labeling is required if the food contains a potential allergen or higher levels of a toxin, although it is unlikely that such products will be commercialized due to lack of consumer acceptance. The European Union, in response to strong consumer opposition to genetically modified foods, has adopted a precautionary approach. Requires that all foods in which more than 0.9% is derived from a genetically engineered organism be labeled. This has made the trade difficult among countries with opposing use of biotechnology. Cultivation of genetically modified crops or farming of transgenic livestock necessarily requires the release of the organism into the environment. Some people are concerned that this may increase the risk of unintended harm to natural organisms in an environment, or for transfer of the transgene to non-transgenically engineered organisms, which could render them weedy or invasive. The vast majority of the transgenic crops currently grown have been engineered to resist predation by specific insects or to tolerate herbicides. They have resulted in substantial reduction in application of agricultural chemicals and have reduced the agricultural footprint on the environment because the BT insecticidal toxins are highly specific for their target insects and are confined to plant tissues. The impact on non-target insects is very low, while the transgenes have been shown to be transferred to non-genetically engineered crops and wild relatives through cross-pollination. The herbicide-tolerant hybrids do not have an advantage in areas where the herbicides are not applied, and insect-resistant hybrids have an advantage only when predation by the specific insect is high. To prevent the spread of the transgene, cultivation of genetically engineered crops should be avoided in areas where sexually compatible non-transgenic crops and wild varieties are found. Biotechnology products and services have enjoyed huge commercial success. Not only have the developers of the technology and products reaped the financial rewards, so have some of the downstream users of the biotechnology products. In particular, farmers in both developed and developing regions who plant genetically modified crops have benefited from increased profits due to higher yields and fewer pesticide applications. However, opponents point out that the cost of biotechnology products is high for both the developer and the users, and this limits the products that are available. On to the review questions. Discuss some of the reasons why foods derived from genetically engineered organisms might be safer for consumption than those developed through selective breedings. Discuss some of the reasons why genetically modified foods might be less safe. What is meant by the term substantial equivalent, and how is it used to assess food derived from genetically engineered organisms? Why are animal feeding trials important in the evaluation of the safety of genetically modified foods? What are some of the benefits of golden rice? Why has commercialization been delayed? How have some reports in the popular media contributed to the opposition to genetically engineered foods among consumers? What is starling corn and why is it no longer marketed? When is a genetically engineered food required to be labeled in the United States and the European Union? Why are the requirements different in the regions? Explain why the risk of incorporation of a transgene in genetically modified food into the genome of the consumer or intestinal bacterium is low. Discuss the potential for cultivation of genetically engineered crops to decrease biodiversity. Why is the risk of harm to non-target insects lower for insect-resistant transgenic crops than for insecticide sprays? Describe some ways in which genetically modified organisms can benefit the environment. How have farmers benefited economically from growing insect-resistant transgenic crops? How have consumer attitudes toward genetically engineered and stringent regulatory requirements limited the development of biotechnology products? How do they impact trade among nations? We're done with the chapters now. I'm just showing you the glossary. So you can just about copy as it's shown. You take screenshots, right? Various terminology is used. adenine adjuvant aerobe affinity purification, affinity tag, AGL fermenter, alternate algorithm alignment. Here the terms were, terminologies will be adapter, isolation, acyl carrier protein, activator site, activator activation, actin acetylation, ATC synthase, oxidase, deaminase, CC absolute linkage, AB. Alkaloid, LL, allelic ferric frequency, allochemical, allergen, allogenic, allolactose, allosteric control, allosteric regulation, alternative splicing, AMP mutation, amino acid site, aminoacyl tRNA, aminoacyl propane, 1-carboxylate amplicon, amylolytic anaerobe analytic probe, and protein microarray annealing. Annotated database, antibiosis, antibiotic, antibody, anticodon, antifreeze protein, antigen, antigenic determinant, anti-idiotype antibody, antiparallel orientation, the arrangement of two strands of duplex DNA molecule which are oriented in opposite directions so that 5' phosphate end of a strand is aligned with the 3' hydroxyl end of the complementary strand. Antisense DNA, sequence of chromosomal DNA that is transcribed to a DNA sequence that is complementary to all or part of a function. Antisense RNA sequence that is complementary to all or part of function RNA. Antisense therapy, the in vivo treatment of a genetic disease by antisense therapy in vivo treatment of genetic disease by blocking translation of a protein with a DNA or RNA sequence. Antiserum aphid, apoplast, set of intercellular spaces that are inside a plant but outside the cells. Apoptosis, controlled process leading to death of the cell that occurs normally during development of a multicellular organism or in response to cell damage or infection, also known as programmed cell death. Aptamer, synthetic nucleic acid, typically 15 to 40 nucleotides long that has highly organized secondary and tertiary structures and binds with high affinity to a protein that normally does not bind to nucleic acid. These are the others. Try to refer as many glossary terms as possible because these will help you in the future of your research. So we're almost at the end of the lecture. And for those of you who need more details on the glossary, you can always write to me. So that ends with the subject of biotechnology. I'll just complete the glossary with a little extra time. right? And then you can copy on, right? You can stop it in between and zoom in and check out the words. I'm just getting it completely copied onto your screens, right? So that you can use them for references. So that's the last zinc finger protein sequence, specific DNA binding proteins that contain domains that bind zinc. Right, those are the reviews, the glossary, and the index. So thank you for listening. Please use all the 23 chapters that with the details of each topic that I mentioned to the best of your preparations. Please take maximum screenshots and develop on your research and notes. Kindly do not commercialize any aspect of these lectures. I'll give you more video links as much as I can on each of the subjects, right? And best wishes. Any doubts, questions, clarifications, or queries, please come out to the comment section on YouTube, right here, or onto my WhatsApp, and I'll guide you through. And additional announcements would be about training classes in OET and relatively IELTS as well. It's available at very affordable rates. We're also training for the prometric dental exams to the GCC. For those of you who'd like to know more details, you may please log and contact me on my WhatsApp number 9188661251. I'm Dr. Jacob KORAH signing off. I'll be back with chemistry very soon for both the AIIMS&NEET and the CSIR Life Sciences. For additional details on life sciences, all right, and other subjects as well. I think you already have a list of the life sciences for CSIR. So I'll be back with more details soon. So take care. Best wishes. Bye.